Hello everyone, today, it's my pleasure to introduce the paper submitted to iMeta. The title is Characterizing the Microbiome of Sterile Organs in Experimental Mice and Evidence of Translocation of Bacteria from the Gut to Other Internal Organs. The internal organs of healthy mammalian animals such as the brain, heart, kidney, liver, and spleen have traditionally been thought to be sterile. Recent studies have challenged this notion, demonstrating the presence of living microorganisms in the organs of both healthy and diseased individuals. In this study, we aim to determine whether microorganisms exist in the sterile organs of the experimental mice. Using culturomics and metagenomics analysis, we demonstrate that over 20% of the 104 analyzed mice harbored a high microbial burden in the internal organs. Metagenomics analysis together with germ-free mouse model assays suggest that microorganisms in the sterile tissues could be associated with the gut microbiota. Given use of experimental mice in medical and biological research, these findings of resident microorganisms in the animal's internal organs raise concerns about potential variability in experimental outcomes. A total of 104 mice, including C57BL6J, BALBC, and ICR, from 3 Chinese providers, WTL, NMO, and ZYU, were tested. All mice were healthy with no observed debilitating phenotypes. Mice were humanely euthanized, strictly disinfected, and dissected under analyzed conditions. The dissected organ tissues, brain, heart, kidney, liver, spleen, lung, were collected from microbiome analyses, including culturomics and metagenomics approaches. Parallel environmental negative controls were performed to rule out contamination originating from the laboratory. Culturomics analysis was performed on all 104 mice using 7 optimal media under both aerobic and anaerobic conditions. Metagenomics analysis was conducted on 10 mice with relatively high microbial abundance identified by culturomics. Culturomics analysis revealed that 23 mice exhibited microbial loads exceeded 1 by 10 to the power of 4 CFU per gram in at least one organ, while 42 mice exceeded 1 by 10 to the power of 3 CFU per gram tissue in at least one organ. A comparative analysis of microbial species detected by both culturomics and metagenomics revealed overlapping species, among which L. murinus exhibited a high relative abundance. The gut of animals harbors a diverse community of microbes. To investigate whether there is a potential link between the gut microbiomes and organ microbiomes, we performed metagenomics analysis on the fecal microbiomes of the 10 mice. We found that, compared to the fecal microbiomes, the microbial profiles of the organs exhibit more similarity. Most of the intermicrobial species could also be found in the fecal microbiota, indicating a potential link to the gut microbiota. Interestingly, L. murinus exhibited a high abundance in both organs and feces. We further re-evaluated L. murinus abundance in the organs and mesenteric lymph nodes, MLNs. We found that L. murinus was detected in MLNs of all the 10 mice, in the brain and lung tissues of 2 mice, in the kidney and spleen of 4 mice, and in the liver of 5 mice. In 8 out of 10 mice, the abundance of L. murinus in the MLN is higher than that in the other organs. These findings suggest translocation of L. murinus from the gut microbiota to MLNs, with subsequent dissemination to historically sterile organs. To further investigate this potential translocation, we performed an oral gavage assay with L. murinus in a germ-free mouse model. 6 germ-free C57BL6J mice were orally fed with L. murinus cells, and the abundance of L. murinus in the different organs and MLNs was examined. L. murinus was detected in at least 2 organs of all treated mice. Consistent with the aforementioned results, L. murinus was detected in MLNs of all the treated mice. In 5 out of 6 mice, the abundance of L. murinus in the MLNs was higher than that in the other sterile organs. L. murinus was the sole bacterium identified in nearly all the organs. These results suggest that L. murinus cells could be first transferred from the gut to MLNs and then to the other organs. In summary, our findings reveal the widespread presence of living microbiomes in the traditionally considered sterile organs of laboratory mice with different genetic backgrounds. Microorganisms found in the sterile tissues are associated with the gut microbiota. The presence and variation of microbes in different mouse individuals may exert physiological effects and experimental outcomes, raising a caveat about the interpretation of results. iMeta, a journal to change the world.